All right. A little review on cellular respiration. All right. So we have glucose outside the cell. So here's our cell membrane. Right, so we have glucose outside the cell. Remember, it comes in. That's a bad P. Becomes glucose 6-phosphate. Remember this process, glycolysis. Right? So process glycolysis makes pyruvate. So makes 2 pyruvates. Now this all happens in the cytoplasm. Right, so the cytoplasm of the cell. When it comes in the cell, the pyruvate it has pyruvate here, but that's not pyruvate. Remember this right here is acetyl coenzyme A. Right, and the acetyl coenzyme A will go through the Krebs cycle. Right, or we'll make things like NADH. And NADH is my electron carrier. So remember, NADH right here is my reduced version. And it's reduced version because it's gained an electron, right? Actually, it's gained 2 electrons, but it's gained electron charge. So we have this system going on here. So remember, glucose came in, became glucose neo-6-phosphate. And through the process of glycolysis, I produced pyruvates. The pyruvates were brought in to the mitochondria and became acetyl coenzyme A through acetylation. Went through the Krebs cycle. I produced CO2, which I exhale. And then my electron carriers become reduced because they gain an electron. Right. So now that all that's happened, I'll switch colors here. So now all this has happened, my electron carriers here will be brought up to the ETS. So my electron carriers here are brought up to the ETS, or ETC, sorry. They're brought up to the ETC. Remember, that's the electron transport chain. And through a series of redox reactions, right? So oops. Right, we pump hydrogen to the inner membrane space, and we're going to use that proton motive force, right, to the inner membrane space, and then we're going to say the use to produce ATP. So how much ATP? Right, 32. Right. All right. So, a couple things when you look at the mitochondria here. Notice how it's shaped. So I've got like a bean-shaped structure, but in it, remember I have the cristae, and the cristae are these folding membranes, the intermitochondrial membrane. So, and in the space of the membrane, or in the membrane itself, you'll find the ETS, right? And that ETS, remember, pumps the hydrogen to the inside, and the hydrogen runs to the outside through ATP synthase, which I'm going to talk about in a second, and it makes ATP. All right, so oxidative phosphorylation, I'll go over this briefly and then we'll be done. So, remember I have the intermembrane space here, and this, right, is the mitochondrial matrix, or the cristae, right? So, this structure right here is that winding structure inside the mitochondria. Okay. So remember, this right here is the ETF, or ETC, or electron transport chain, right? And these are my electron carriers were fed in. Oh, there we go. My electron carriers were fed in. So important to remember that these hydrogens will now get fed through ATP synthase, right? And that's going to produce ADP plus PI. It's going to become ATP. All right. So a couple important things. Remember, this is oxidative phosphorylation. So O2 is going to come in. And this is going to take my spent H plus, and I'm going to make water. All right. So I'm going to make water out of O2. So O2, you remember here, that's going to be my final. My final electron acceptor. Okay? Oh, it does it. All right. Now, important, one last thing. So my ATP here, so my ATP synthase, right, is an enzyme. All right. So remember, an enzyme is a protein, and the protein is made of amino acids, right? So I have my polypeptide. Remember, that's my primary structure. I have my alpha helix. My beta pleats, right? And that's my secondary structure. I have my conformation, which is my tertiary structure, right? And this conformation right gives a function. So if I heat this up, or so my conformation is my 3D structure, and it'll have something called my active group. Remember, that's where substrates fit. And if I heat it up, right, or I cool it down, or I make it too acidic or too basic, it changes the shape of that active group and it doesn't fit anymore. Okay, well that should be all that you need to get through your free response questions for the beginning of this week. All right, I hope you had a good week. You're all staying safe and well, and I will talk to you soon. Bye.